Besides the use of bone marrow stem cells for treating hematological malignancies, which is an established clinical practice today, bone marrow cells are also under intense investigation for regenerating various organs such as the heart, the liver, the lung, and so on. In this particular study that we're talking about today, the possibility of using bone marrow to treat the disease ALS is investigated. ALS is a lethal condition associated with the degeneration of motor neurons, the motor neurons in the spinal cord, cerebral cortex, and in the brainstem. The actual cause of ALS is unknown, although some patients have a genetic mutation that seems to correlate with ALS, which we'll touch upon in the next slide. And at present there is no cure for ALS. The question of the study is whether bone marrow cells from healthy mice can inhibit the progression of disease in a mouse model of ALS. And subsequent questions, of course, include whether the stem cells transferred, whether they can actually go across the blood-brain barrier. So the model system that was used to ask this question involves a mouse which displays a predisposition to an ALS-like disease. How this mouse was created was that the human mutation, it's called an SOD, glycine 93 to alanine mutation. SOD is an enzyme called superoxide dismutase. Now when you take this mutated enzyme and you make mice to express this enzyme, the mice end up developing a pathology like human ALS. So what the study did was it transferred bone marrow from healthy mice into mice that are predisposed to ALS to try to see if the process of progression to ALS can be inhibited. Now, in order to track the cells of the healthy mice that are put in the mice which will develop ALS, GFP mice were used and YFP mice. What this means is both GFP and YFP mice are healthy mice. The only difference between a normal mouse and a GFP and YFP mouse is that the GFP expresses the GFP protein. It's a green protein that lets you track the cells. YFP is a yellow protein which the mice were generated as such so that YFP, yellow, will only be expressed in neurons. And these cells, the healthy cells, were administered, 30 million of them were administered after irradiation of the recipient predisposed mice, in order so that the cells administered will mimic the administration of cells in the context of a bone marrow transplant. So, if one looks at survival, the graph here demonstrates time on the x-axis, and on the y-axis is the percent survival. And as one can see, the blue line and the red line represent survival of mice which received bone marrow. The red line is from GFP mice, and the blue line is from YFP mice. So mice that are predisposed to become sick that received labeled bone marrow live about 20 days longer, as opposed to mice, the black line being mice that received bone marrow from another mouse that is predisposed to become sick, and the green line is a mouse that is untreated, is the group of mice that are untreated. So this figure tells us that the transplantation of bone marrow can, statistically significant manner, increase survival. The next question is about functionality. So here the GFP bone marrow was transferred into mice which are predisposed to ALS, and this is illustrated in the red line. The green line is control mice, and as you can see, the y-axis is coordination ability on a test called the rotarod, and the x-axis is the time, the days. And as you can see, although both mice do undergo a deterioration of coordination ability, the deterioration is markedly inhibited in the mice that receive the bone marrow. Is there any evidence now that the cells are actually differentiating or contributing to slowing down deterioration at a cellular level? In this figure, one can see that wild-type mice, not mutant but regular mice, they have a certain number of motor neurons. This is basically a microscopy. Um, the lumbar spine was taken from the mice, and it was assessed under microscopy, and the number of neurons were counted. And as you can see on the left, the wild-type mice had an average of about 25 neurons per viewing field. The mice on the right-hand side, the SOD transplanted or the SOD non-transplanted mice, they had an average of about 11 neurons. And the mice that received healthy bone marrow, GFP or YFP bone marrow, had a statistically significantly higher amount of neurons than the control mice, indicating that there is actually a preservation of neuronal content in the lumbar spinal cord and also in the ventral root axons. Now, when one actually looks at the recipient mice, and bear in mind again that the recipient mice should have no green cells. When the recipient mice receive GFP bone marrow, one can see neural differentiation. This is a neuronal shape, and these are cells, green bone marrow cells, in the recipient mouse, having differentiated into what appears morphologically to be a neuron. Now, one can also look more specifically at types of neurons that were developed. Here is looking at Purkinje cells. Now, the Purkinje cell expresses a marker called calbindin D28K, and the green cells, of course, are just GFP. This is from the donor mouse, where all the cells are green, into the recipient mouse, where there's no green cells. So in the A, you can see what appears to be a Purkinje cell based on morphology, a green Purkinje cell, meaning that the bone marrow differentiated. In B, you can see staining with the calbindin marker, and as you can see, there's 3 red cells there. Now, when you overlap the staining, you can see, which is in C, you can see that there's 2 red cells on the side, which are presumably host-derived because they do not have any green, and the cell in the middle has green, implying that it differentiated. That is a bone marrow cell from the GFP donor mice that differentiated into a Purkinje cell. The same can also be seen with glial cells, where green is a GFP, meaning it's from the GFP donor. Red is a stain for glial cells, F480, and as you can see, there is double staining. So in conclusion, the study seems to suggest that bone marrow stem cell transplantation has a positive effect on the progression of ALS. It also demonstrates in a very black and white manner that the cells do cross the blood-brain barrier. The study suggested that there's numerous donor microglial cells that cross, and we didn't discuss numbers, but the study did mention that there's a lot less neural differentiation and as opposed to microglial differentiation. And also the study mentioned that fusion between the donor cells or recipient cells seems to be associated with neural differentiation. Now, what are the next steps? And remember, this field has moved because this was in 2004 when it was published. Next steps is we need to ask, do we really need complete chimerism with a donor to see the effect? As we talked about, when you irradiate the mouse, it's similar like in a bone marrow transplant situation, which is associated with significant mortality and morbidity. We don't believe that you need complete chimerism. Also, other types of stem cell population should be investigated, such as cord blood cells. And because ALS is a multifactorial disease, there's a lot of rationale suggesting that stem cell therapy may synergize with existing and experimental anti-excitotoxic modalities. Thank you very much.